Albany to kick off today's talk. And then we have Catharine, that's from the Functional Genomics Center at Zurich. I'm from Olink. The reason I'm up here today is just to introduce real quick, but there's a program also tomorrow that did not make it on the schedule, and that's by Graham Wiley at OMRF. It's somebody that runs a genomics core, but has also stepped into proteomics, hence from Olink. So thank you very much for coming today, and I'll let the speakers take it off.
It's all right. Well, this should be, go to the next one. Nope, that's not me. Nope. It should be up there, that one. Yep, that one. There we go. Got it. So, perfect. Good evening, everyone. I know we are the last session before you guys decide to go out and do your Las Vegas shenanigans. So we won't keep you long. I'm Sridar Chittur, co-chair of the Genomics Research Group, along with Catharine Aquino Fournier. So today, there are two parts to our session. The first part, I'm going to talk to you some of the preliminary results that we have from our survey. So, and Kathy is going to be talking about the partnerships at her core between genomics and proteomics cores. So, before I start, we did a survey. I don't know how many of you got to it. If you haven't taken the survey, please scan the QR code and take the survey. The goals of the survey were to identify the approaches used by different labs to quantify the samples for pooling, identify the challenges in preparing a pool that permits getting the best the coverage for each sample in a cost-effective manner. And then based on these results, we are going to plan our study for next year, right? And who cares about pooling, right? Anyone? What do you care about pooling? Yeah.
Because if you don't pool it right, you're wasting money.
Right. But why do you even need to pool?
Because if you don't pool, you're wasting money.
Right. Everybody is buying these bigger and better instruments, and the challenge is we have a lot of capacity in our course, right? Those instruments are not running 24/7 packed to the brim, and that makes running each flow cell expensive. We also are mostly from scientific core labs, which means we don't know what's walking into our door tomorrow. Someone's bringing metagenomic samples, someone's bringing whole genome samples, someone's doing amplicon sequencing, and everybody wants the data yesterday. Right? So there are two aspects of it. We want to batch everything together so we can load our flow cells, and we also want to be able to do these experiments quickly so we can deliver to the end user. So that's why we started with this survey. As of last week, we had about 58 responses. And as you can see, we have a lot of them from the US, but we had a few from Europe as well. Most of our responders were ABRF members. And for the people who are not ABRF members, I would suggest you should consider becoming an ABRF member, because hopefully you realize that this meeting has a lot of different things to offer, especially for shared research labs, right? Whether it's from the wet bench side, the bioinformatics side, the administration side, there are different things that you can learn here, because misery loves company, and we like to share our stories, and there's no need to reinvent the wheel if somebody's already done it. And this community is quite different from the typical scientific communities where everybody is competing with each other. Here we are willing to help each other out and share our best and worst experiences, right? So, most of the responders were academic. There were a few industry responders, and there was one government responder. as well. So the first question was, what type of instruments do you have? Well, surprise, surprise, most of the users are Illumina users, so short reads. There are a few element and singular with the new short read sequences that were introduced a couple of years ago. So now we have seen a few people who are doing element, singular, and then we had the long read with PacBoy and Nanopore. We have not had anybody respond yet who's had a complete genomic system. I know that Complete Genomics was placing these in New York, but I'm surprised no one has responded. They just haven't gotten to the survey yet. And yesterday I was at the talk in Element, and Minnesota has an Ultima Genomics. So I told Jerry, please take the survey, right? So hopefully the goal is we'll take these, we'll keep collecting these data, and by the end of summer or early fall, we can pull these things together and publish in JBT so it's accessible to everybody. I mean, sample size, right? We all want good sample sizes. Okay, so based on the sequencing results, the sequencing instruments, you had people running anywhere from the MySeqs, the NextSeqs, both versions, the NovaSeqs, the AVTs, and the singular instruments. And on the long read, you had the PacBio Reviews, the MinIONs, GridION, Promethean, what have you. And as you can see, the MySeq users were by far the largest. Now, I have to say that if you took this survey, you would realize that you could choose more than one. So these are not exclusive, right? So there are many people who have a MySeq and a NovaSeq and a NextSeq, so there is a lot of overlap, and I haven't quite parsed that out. And there are lots of labs who have actually been using the short read platforms and are getting into the long read platforms, and there are some going the other way too. In terms of library or sample QC, whether you demand that every sample that walks into your door has to be QC'd, or do you accept the the QC supplied by the user, or you don't care, you will run everything. Most of the people, 70% of the users require that each sample that walks into the core has to be QC'd by the lab. Right? Now, about 30% of the course said that they would waive the QC if the user is willing to accept the risk and pay the bill at the end of the day. Then they'll run it. And as core facility staff, we all know what that means. It's like sometimes you can tell them the sample is not good, it's not going to work, don't run it, go get a different one or what have you. Now, I want it run. Okay, as long as you're willing to pay for it, we'll run it. We don't control much of that, but we do. If the data is bad, don't associate our reputation with that. So in some labs have a way of QCing just a subset of the samples to a quick batch analysis, and if it works, great, we'll run all the samples. Some of them, based on the long-term experience that they have with that particular PI's lab, they might decide whether it needs to be QC'd or not. I mean, everything that's been sent by this lab is great, so and the lab, the PI is easy to work with. They've been following the best practices generally, and so there isn't much of an issue that we expect. There's some of those. And then some users don't have access to these things, and so they bring it to the lab and say, make sure that you do it. And then there was one person who said we QC all the libraries, but if the user submits it, we'll accept it, but we don't necessarily do the samples. And there's another one that said that we'll be required QC by the law for each library, but not the samples. But for the most part, the lab requires that QC metrics to be done in that lab, right? So in the core facility. The types of libraries that were processed, well, anywhere from RNA-seq, ATAC-seq, whole genomes, amplicons, microbiomics, metagenomics, DNA, RNA methylation, proteomics, which is up and coming, single cell, spatial, isoseq, and then some cut and tag and chip and run. And as you can see, single cell is pretty much there with RNA-seq. Those are the two technologies that are being widely run in these cores. Okay. So how do you QC the libraries? Well, you could use a picogreen or qubit or something like that. You could use a nanodrop. there are uses to it, even though it's not going to be good for sequencing. Capillary electrophoresis, where you could be using the a good old bioanalyzer, the tape station, the fragment analyzer, the femtopulse, what have you, your favorite. All of those fall under capillary analysis, capillary electrophoresis. You could do qPCR, NEB, kappa, your favorite method, or digital PCR. And most of the labs use these three methods. So they either do a combination of a qubit, fragment analysis, and then qPCR. And why do we need to do that? Most of the people here should know that answer. But in the case, if you're new to NextGen or if you're new to working in a core, just so that you don't get blamed, you need to make sure that you set yourself for success. So this will give you your DNA concentration. This will give you the size of your libraries, and so you can see if there are contaminating small fragments, which can cause issues, or really long fragments, which can also cause issues. And then qPCR to make sure the adapters are on that insert that you're going to sequence. So if you're missing those, yeah, you put the DNA in, but you're not getting the reads out. So the other question we asked is, how do you implement the standard operating procedures and protocols for your lab work? Do you create SOPs based on the vendor recommendations, or do you make your own? Do you do automation? Do you create your own SOPs regularly, irrespective of what the vendor says? Do you strictly follow whatever is in the manual? Or you don't have any SOP and you're freestyling it? And as you can see, most of the labs are doing SOPs based on a combination of what's in the manual and tailored to that particular project or those types of samples. Because the vendors can't test every type of sample or every condition. So, and labs also have to look at the economies of those products, and so you might mix and match different things to make it the best kit that is economically feasible in your hands, in your lab, for your type of samples. Then, of course, with automation, you got about 25 people of those 58, so half of them that are doing automation. A third of them are doing creating their own SOPs, and then. Now, do you pull different types of libraries, i.e., whether it's DNA, amplicons, whole genomes, RNA-seq, metagenomics, and your flowcell? Yes or no, right? And you can see that most of these people, it was half, I would say. So half of these people said yes, half of them said no. And so when you look at why are you not pulling your libraries, well, the question was the risk of bad library inhibiting the other. So if you have short libraries and you have a long library and you're trying to run them together, you could have one preferentially being sequenced over the other, and so you get more reads of these that you don't need. You have a possibility of index hopping on some platforms. It's just too much labor, because now you have to see whose samples, which PI, what have you, make sure that you're keeping track of all of those things, and the blame's going to be on you if something goes wrong. Too much of a hassle. On the X Plus, people said that there was variable clustering, and then library size can also impact that clustering. Then different labs preparing their own libraries, sending it to you. If you are doing everything great, you control the whole process. If you're getting different samples, different libraries from different labs, the indices that they use, the size of the indices, whether it's a 6 barcode, 8 barcode, 10 barcode, whether they're UMIs or not, whether it's single index, double index, it's all over the place. So trying to batch those together is also challenging. And then there was one person who said, we have a G4 and it's got 4 independent lanes, so we don't need to pull different libraries. We can run them each type of experiment on a single line. What are your considerations and concerns when you're pulling the libraries? Well, if you do pull the libraries, then the biggest thing is the fragment size. You want to make sure your libraries are all of the same size. The protocols that you're using are also need to be looked at, whether you're doing amplicon sequencing versus 16S amplicons or highly repetitive amplicons with highly repetitive sequence, or what have you, depending on your protocols, right? So, and then the primer dimer content of your adapters, your barcodes, this is the big thing. So make sure that, at the end of the day, you need to know which read came from which sample. So if this is messy, then all bets are off. Reads per library, do I need to have everything give me the same amount of library, or can I spike in a little bit of something and more of the other, so that this one I only need 100 million reads, that one I need 400 million reads, or what have you, so I can control the ratios of the samples that are spiked in. Do I pool everything with unique barcodes? So we don't care, as long as I can tell them apart, I'll pool everything. They were 8 people who said they would do everything. What is the maximum libraries you pull on a single sequencing lane? Well, for MySeq, it went all the way up to 384 amplicons. For the next seeks, whether you're running the 2000 or the 6000, it was 400 amplicons, about 30 to 60 RNA-seq. For the larger instruments, it was people are mixing RNA-seq, whole genome, single cell, all of these experiments together. The NovaSeq, of course, the big gorilla, it can handle a lot more samples. On the element and the singular, it's flexible, but again, the element is the people who reported were doing up to 384 amplicons. I'm sure that there's more of this that can go on the element. So, but survey responders haven't said that yet. So, if you haven't taken the survey and you're doing something different, let us know. That'd be very informative to the group. On a PacBio, about less than 20 samples, and on the Oxford, about 48 to 96 samples are being pulled. So one person said, we'll pull as many libraries as necessary as long as they're compatible. The yield per library can be achieved with the flow type run. And they're primarily limited by the available indexes. And this is true especially for things like the Oxford, where you don't really have a 384 barcode or a 1500 barcode set that you could use with your RBK kit and run with it. So there's some room for development in case the vendors are listening. How many reads per flow cell do you cushion typically as do you typically target under the maximum flow cell output rating? So what that means is if my flow cell is supposed to give me 400 million reads, do I tell my user, okay, we are going to give you 400 million reads, or are you telling them I'll give you 350 and then you have that cushion, so in case, no one's going to complain if they have more data, but they will always complain if they have less data. So are you giving yourself that cushioning? And sorry. If the libraries that are being pooled are of the same library type, then there are people who, there are about 4% of the people who are getting about 25% cushions. That's like a quarter of your flow cell. Well, most of the people are using about a 10% cushion, which seems to be reasonable. I mean, we do that too. Between 5 and 10% is what we sort of reserve and keep. And if you're mixing different library types, then again, it's about the same. About 30% we're using about 10% cushions. But then you have some other flavors because now you've got different types of libraries and the expected amount of reads per type of experiment is slightly different. How do you normalize your libraries, where some of them spike an aliquot into another run and see if it's working and get your reads from that. Some actually do a low throughput NGS run on an aliquot of the sample. So they could do an iSeq run or something before they throw it on an OSSEQ. Some do enzymatic normalization, but for the most part, people are using capillary electrophoresis and qubits. And then normalizing qPCR or digital PCR and the standard NEB or kappa qPCRs. Do you physically pull your samples? Do you ask your users to pull the samples? Do you use robotics? That was another question. And when you're pooling, do you create a subset of pools while making your, if you're pulling 384 samples, I mean, are you actually pulling all 384 of them, or are you making subsets of them, pools and then pulling the pools? Right? So in just that, if some mistake happens, it's not throwing all of them at the same time. So for the first question, looks like most of the people so far are doing mostly hand pipetting. There are some people who are using the robotics, and 6 people are using both the robots and the hand pipetting. Of the robots, the ones that were reported were the Janus Cyclone, the Hamilton, the Opentrons, the Echo 525, the Revity instruments. The Myra, I hadn't heard about it till I saw the survey results. Do you create a series of subset pool, a series of subpools? Well, it's about a third of everything. Yes, no, sometimes, depending on the project. So that wasn't very informative. Now, this was an interesting question. How much do you estimate in US dollars? How much revenue you've lost because of inaccurate pooling, right? Including consumables, labors, and opportunity costs. So most of the people are saying about $5,000 is what they're losing in a year for bad experiments or bad pooling. Another 6 of the 56 people were about between 5 and 10. But there was 4 people who said they're losing 25 to 50. So these must be the NovaSeq or X Plus users where something went really bad. And just those flow cells cost a lot. So that's money down the drain, and especially in this environment when you don't know if your PIs are going to have their grants and whether your cores are going to have the money in the budgets to run. I mean, this is something that one really has to watch out for. So again, we hope that with more input, we can rationalize and make some best practice recommendations what you could be doing, and hopefully that will guide our next year's study as well. Questions? That's Luke. So he's my favorite child. And he's missing me and I'm missing him.
Did you scale the revenue lost based on exponential type or anything? Because it seems like it would be highly dependent on what sort of platforms the lab would have.
Right. Again, we have to look at those more granularity, with more granularity. And so, we need more. It's only 58 responders to begin with. I need to look at those. And this was a quick survey, unlike the other DSRG survey, which we had a lot of questions. This was a quick and dirty one, because we want to see are people interested? Is this something that, the reason we did the survey is, to help educate ourselves as if you're not polling, why aren't you polling? Because you need to be thinking about that. If you're polling, are you doing the right things? Or looking for things where you could improve your best practices. But on the other hand, what are we, if there are challenges that people are pointing out here, then that is something that as GRG, we can use as part of a study and tackle that and come up with better methods, wet bench methods downstream. So, but good question, Sarah. Good. Ioannis?
So it's super interesting. You had this slide where you showed the library types and how many people are doing this. I didn't ask you to go because it's quite archaic. Yeah, this one. But it would have been interesting to see the volume for this, because there are bigger libraries and smaller libraries, and you could be doing this all the time or sometimes. I know it's a lot of granularity, and if we ask for it, often people will not go into that detail. But it would be interesting to see that scale on volume, sequencing volume, but also the number of libraries as well. Because the effect of pooling is different.
It's going to be a huge skew with mid-sized labs and larger labs that's doing whole genome sequencing. I do virtually none of that on Pac5.
Bless you. Okay, no other questions, and we'll have our next speaker, Kathy, come up and talk about her experiences in
Clicker doesn't work. Now you can. Now you really can't see me, I guess. No, it's okay. But we can just put it there, maybe, because then the mic is. It's always a problem. Sorry. But now I'm far from the mic. Okay. You can hear me, no? Yeah. Okay. I have only technical issues today, also for with the lightning talk. So anyway, so when we were discussing with the GRG what else we can present during this session, we then thought that to present something about the new methods like NGS-based proteomics, because apparently that's a little bit complicated, because now you're dealing with a few different units that has to interact together and work together, and obviously there are other challenges there as well in the implementation of these workflows. And at that time, we were testing the Olink reveal last year. So I was very brave to say I will do something to talk about our experience with the Olink reveal and how we interacted with our proteomics unit. So the title of the talk is Implementing NGS-based Olink Technology for Plasma and Serum Proteomics. The experience of Functional Genomics Center in Zurich. So we're a core facility of two universities, the ETH Zurich and the University of Zurich, and we have been doing multiomics since 2002. So we have genomics, proteomics, metabolomics, and bioinformatics. And we are all happily living in one roof. Because we have a good coffee machine. So there are challenges, a lot of challenges apparently. So I just also want to say that this is from my point of view, so the genomics unit point of view. Our proteomics unit will present their side of the story at ASMS. But maybe show of hands first, who is a proteomics person here? Okay, not so many. Very good. So I will be more biased towards our experience as a genomics unit. Anyway, so obviously I'm not a proteomics person. Okay, clear? So liquid biopsies apparently is very challenging to do, and this is one of the in proteomics, and it's one of the difficult samples in clinical proteomics. And the gold standard for analyzing these samples are LC, liquid chromatography, mass spectrometry methods. But it requires, of course, a lot of equipment and expert knowledge. And also, what is also important is the complexity and the dynamic range of these samples are very challenging for mass spec-based proteomics. And so then we have been discussing with our proteomics team for a few years now if we would actually implement the Olink or something else, homologic, Alamar. We were looking at everything, but the problem with all the other workflows that we looked at is the CapEx. Because for example, with the Olink big panels, we have to buy, dedicated instrumentation, and that means that we really have to process a lot of samples. Otherwise, it would be a bit of a waste if we were just processing, 100 samples per year. And then, so when last year Olink was approaching us if we wanted to test the reveal, and then because of that there is no CapEx, then our proteomics unit actually agreed to do it, which they were not agreeing to do before with the other samples, with the other methods. Yeah, so what we tried to do is we tried to see, because obviously they still needed convincing our proteomics unit, so we wanted to check if it's, the biological relevance of the data produced with Olink compared with their LC-MS method that they trust. So I think everybody knows that Olink reveal. So it's a targeted version of proteomics. There are only around 1,000 proteins. It's still antibody-based. And as I said, what's nice about it is there is no CapEx. The workflow is very fast, so I did it myself, and I have not pipeted for like a year or more, and so I did it in a day and a half. So it's very quick, and so basically it's just an incubation step, 16 hours, and then a PCR, and then a cleanup, and then you sequence, and then it's done. So the whole thing was we sequenced it in 3 days. So after so the whole workflow took 3 days. So, with the same set of samples, then we did two kinds of, so our proteomics unit did two kinds of methods from LC-MS. So this urea in solution digestion, and enrich plus. So basically, it's more like an enrichment-based method. Yeah, and then they loaded it on these two instruments. So just to say, I was very proud of myself because when I was doing these slides, I actually chose the right instrument and it was correct. So this is EvoSep and something else. And yeah, so they have a lot of pictures in our website and I was choosing the right one, so I was very proud. I don't know. What is it, Ralph? The small one? Yeah, I don't know. Something. Yeah, so our preliminary results, it's very preliminary because, okay, there are only 2 proteomics people here. Because it took them 3 months to generate the mass spec data. So that's why we are still analyzing the data. So I thought I would have more data by now. And then we found that actually, so based on our results, it seems like these are orthogonal approaches and the overlap is very small. It could be really because of biology. It's just because, you know, when you do the without enrichment, so you also see more the abundant proteins. And then the enrichment based method, you also see something else because it's like you just take out the abundant proteins and then look at what's left. So basically, there is very little overlap between the 3 methods. And then we're still checking which is actually, correct from the samples that we have processed. So for the workflow comparison, so what is interesting as well with this Olink reveal is it's not very expensive. So it's like similar to an RNA-seq with pricing. So basically, it's with sequencing, it's about 150 francs, which is $150, or depending on the exchange rate. And then obviously the throughput is very different because with the Olink reveal, basically you can do as many plates as you have PCR machines. And then for the urea-based method, it's about 1 plate a day. And then for the enrichment-based method on mass spec, then it's around half a plate per day. And then obviously the number of proteins that we can find is also quite similar, and the reveal is like in the middle in terms of number of proteins found. So how do we handle these projects? I think this is this is quite important because we operate based on the molecules that we are, the unit is fond of. So this kinds of project is still a project of the proteomics unit. So they will decide. So they have the contact with the users, and then they coach the project, and then they decide later if it would go on the LCMS or they will be our user because they will forward the samples to us, and we make the libraries and the sequencing. Or, because also the method is quite easy, our users, the 2 projects that we have discussed recently, it's basically they just want to do the samples, they just want to process the libraries themselves and then just send us the samples for sequencing. So basically also bypassing the proteomics unit. And how do we handle the data? So we handle the data based with our, I think I did like 2 presentations already in like a few years ago about our fantastic LIMS system. So we handle this via our fantastic LIMS system called vFabric. So this is able to basically accommodate multiomics data in one container. So as you can see here, you can in one container, then there is the NGS data, proteomics data, and then everyone involved in the project can access this data, and all the work units or the resources are all in one folder. So in summary for the results, so basically it showed that the reveal and the urea-assisted LCMS achieve the most reproducible results. The Enrich Plus, I think, according to our proteomics unit, but I'm not supposed to say, is not very reproducible. So maybe they will repeat it again, the analysis. So this is a very interesting study for us because it showed like how we can choreograph the projects and how the projects can be a proteomics project going to the genomics using the genomics instrumentation. Some insights, so it can be challenging to do it because I think the proteomics unit and the genomics unit, we work very differently. So for example, I think our genomics bioinformatics is more comfortable with the data because it looks like RNA-seq. But the proteomics people, they're not comfortable with the data because it looks like RNA-seq. So they're like thinking like, why is there like reads everywhere? There's not negative there's not a sample that doesn't have reads. So they don't understand that, or they have a problem accepting that. But the data basically is handled by the two units because basically it uses the genomics infrastructure. So the demultiplexing and so on. So basically we demultiplex it and then send it to our proteomics bioinformatics team. And what else is a bit challenging is that it's a kit. I think proteomics is not very keen on using kits because they're more into doing methodologies, implementing methodologies. And then when it's a kit, it's like, then what are we here for? It's a kit, And I think that's one of the acceptance issues. And also, I think our proteomics is still a little bit shocked about the consumable prices of genomics. So we're always making fun that the whole budget of proteomics is consumable budget. They have like 15 mass specs, I think. And their consumable budget is equivalent to 1 NovoSeq X run. And not even the 25B. Okay, it's an exaggeration, because actually it's 3 NovoSeq X runs. And then we are now in run 270, so you can imagine the difference of consumables from the two units. So, yeah. Anyway, so that's us, and thank you very much. And thank you also to my colleagues who are not here. Questions? I don't know.
Can you give an additional impression about the sensitivity for, in liquid biopsies? It's all about how sensitive can you be for a disease biomarker. So
for which method?
For the OLE.
Yeah, so we don't know yet because we're still analyzing the data. So now we just have this proteins, and then we're still trying to see like which pathway is and then what is actually if it makes sense because with the treatments of these samples that we have done. So we have done like 300 samples, I think. Yes? Okay.
I have a question for you. So did I understand correctly that the results that you got from LCMS and from genomics, or from NGS-based proteomics, didn't overlap? So
no. So the enrichment, it's only the enrichment that overlapped, and out of only 138 proteins were found by these two methods. Yes.
So as providers of NGS-based proteomics technologies, do we need to do something more to make sure that, I mean, maybe with the samples, whatever, who knows what the study was, right? But do you think how do we handle this reticence from not reticence?
Yeah, there is resistance.
To adoption of NGS-based proteomics?
I think more benchmarking studies, because from what I understand, there was not a lot of benchmarking studies like comparing mass spec to SomaLogic in your case, right? So that would be nice, just to see like what do we have here, right? Because every method has their limitations. For example, the one without enrichment in LCMS. So they find very few proteins and that's the most abundant ones. right? And then the enrichment method, there's also a bit of limitations because apparently it was not so reproducible. And also it just also depletes the abundant ones and then keep the rest. And because maybe it's not reproducible because these are patient samples, so maybe it's like real biology, but we don't know. So obviously the antibody-based method, it's very targeted, so you're just really looking at what you want to see, you know, and the problem is you cannot even find it. You know, like with LC, what I understand of proteomics, correct me if I'm wrong, it's really hard to find, right? The not abundant proteins. I think that is the problem with this dynamic range, because you're just getting the, yeah. So I don't know how to, basically I guess you just have to look at the pathway and what makes sense with the treatments or whatever you're looking at, yeah.
When you look at the proteins, there's so many post-translational modifications that happen, which you're not going to capture by any of these methods unless you have an antibody that was detecting each one of those modifications, right? So that could partially explain the differences too. Whereas by mass spec, if it's there, it's going to fly, it's going to hit the target, you're going to detect it. So.
Yeah, but you only detect the abundant ones.
Right.
That's
with
also.
the enrichment, you're sort of like a riboreduction, right? So
Right,
that's what
right.
we're doing.
Yeah, I guess it goes back to the benchmarking part. I guess our community we need to do more of that so that it's easier to migrate through different data types.
Or you know, like, oh, you get what you really can see or what each method really does, and if it's not contradictory, you know.
Yeah, so there's actually a nice paper now on the med archives from Alchemist. They compare different MS methods and then Olink and Somologic, even between mass spec methods.
Ah,
It's not always that.
and it also doesn't correspond, right? Bonnie.
Actually, I was just curious about the scale of operation. So, like, what I understand that you need to run 88 samples at least to get the economy of scale. But each kit can be used only once, and they have to use at once.
The reveal?
You need 88 samples.
Yeah, because they have blanks and then there's controls and
But
so.
you need 88 samples. So, how do you see this possible for a core facility to always to have that number of samples for each project? Or if not, then what strategy you think that you can?
Well, I guess you have to feed that back to Olink to make 48 samples.
No, but at this level, like, how do you see, like, operationally, like, because I think you tested it, so you want to operationalize it.
Yeah, because you buy per plate, right? So I guess you know, if you have 88 samples, then you can say 88 divided by.
My problem is not that you have 88 samples. My problem is like if I don't have 88 samples. So very less possibility, like every project I will have that many samples. I mean, most often I will have the same number of samples than that. So
No, I agree.
what is your strategy and what do you think about
No,
that?
I totally agree with you. Actually, what I also was telling Kurhan, the product manager, is what would be nice as well is to have custom, you know, to be able to add custom proteins. Because I'm sure like people have favorite proteins that they want to look at, not only the 1001, right? So that would be nice. So but I agree, because you have to put 88 samples there. But usually these things are, you have enough. I mean, for example, we didn't have really much problem to get that many samples. Yeah. But that's just how they pack it, but it's not me. Any more questions? Thank you.
If you are not a GRG member and interested in becoming a GRG member, just send an email to Catharine and me, or any of the other GRG members for that matter. Because we'd love to have more people, right? Because doing a benchmark study properly requires more people. I may have one system, you may have a different system, but if you can take the same sample through different approaches and collectively look at it, which is what we've done in the past with single cell, so it helps the field grow better, to have done best practices. The other question I have is, we talked about pooling strategies and we talked about the Olink. Again, these are things that we thought about as possible future studies. If you have some questions that are burning, and maybe this would be, I would really be really cool if I had an answer for this, and this might make a good, send an email. Even if you're not a GRG member, send an email to somebody on GRG, or to the EB or Ken Shopman if you don't find anybody, right? it'll get to us. Because we would rather do something that benefits our community than just 6, 7 people deciding, okay, let's do this. Right?
And one more thing to convince you to join us is we don't do so many meetings. Because I'm bad at calling meetings. But it's efficient.
Thanks, everybody.
Thanks.